The broadcast is now starting. All attendees are in listen-only mode.
Hello everyone, and thank you for joining us today. You have logged on to the Nelson Labs webinar, Fundamentals of Disinfectant Efficacy. Just a few housekeeping items before we get started. If you miss one of our webinars or would simply like to refer back to it, you can always find them on the Nelson Labs website on the on-demand webinars page listed under Education. You can receive notifications for upcoming live and on-demand webinars by following us on LinkedIn or liking Nelson Labs on Facebook. We would also like to invite you to register for free for the Sotera Health Academy at soterahealth.com/academy. There you will find curated content from industry thought leaders, from introductory sterilization and lab testing topics to advanced in-depth learnings. Our expert advisors have filled this academy with cutting-edge educational content and resources to help you mitigate risk, go to market faster, and achieve excellence in your field. Nelson Labs is a leading global provider of laboratory and expert advisory services for medtech and pharmaceutical companies. The company performs over 900 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and protective barrier industries. The experts at Nelson know every test matters and requires solutions to complex problems to improve patient outcomes and minimize client risk. So let's get started. Today we are joined by Cassidy Law and Shelby Quiggins. Cassidy has over 9 years of laboratory experience specializing in microbiology and disinfectant efficacy testing. She began her career in clinical laboratories focusing on genetic sequencing and fragment analysis for 5 years before joining Nelson Labs, where she has worked for the past 4.5 years. Shelby has been a part of the Nelson Labs team for over 3 years as a laboratory analyst and study director. Prior to Nelson Labs, he earned a BS in biology from Arkansas Tech University while being a student athlete on the baseball team. Shelby has contributed within the routine chemistry and microbiology teams, overseeing testing focused on medical device cleanliness and disinfection efficacy. In his current role, Shelby has gained experience in coordinating and guiding a variety of pharmaceutical manufacturers through their disinfectant efficacy validations. We do encourage your audience questions, and you can submit them at any time in the questions box. Cassidy and Shelby will try to answer as many questions as possible during our Q&A portion. Also joining Cassidy and Shelby for the Q&A will be Tanya Morris, Global Segment Leader, Aaron Zani, Expert Consultant, and Michelle Lee, Director of Scientific Improvement. So now to begin today's webinar, I'd like to turn the time over to Cassidy and Shelby.
Thank you, Mike, for the introduction. And welcome everybody to our webinar. As many of you know, disinfectants must be qualified on actual surfaces used within the facility before they can be approved for routine use. This step is not just best practice, it's essential to meet regulatory expectation and for ensuring your contamination control strategy is truly effective. A major part of success in disinfectant qualification lies in how the study is designed. Incorporating information from your in-house environmental monitoring program and being aware of common study pitfalls can make the difference between a smooth regulatory review and receiving unexpected time-consuming feedback. This presentation will outline the key objectives of disinfectant efficacy testing, clarify how it connects to your environmental monitoring data, and give you practical guidance on developing coupon studies that accurately reflect your facility's processes, surfaces, and microbial challenges. Finally, we'll discuss how to interpret and leverage disinfectant study results to strengthen your overall contamination control strategy and ensure your program aligns with regulatory expectations. Before we dive deeper, I want to highlight the key takeaways you should expect from today's session. First, we'll talk about how to choose study parameters that are truly representative of your facility and also meet current regulatory expectations. This is an area where facilities often either under-design or over-design their studies, so we'll walk through what a correct size study looks like. Next, we'll look at how your environmental monitoring program can directly inform the scope of your disinfectant efficacy study. We'll also discuss how to recognize when you need to perform disinfectant efficacy testing, whether it's an initial qualification of a disinfectant, a subsequent study triggered by changes in the facility, or shifts in contamination patterns. Finally, we'll walk through how microorganism log reductions are obtained and how they will apply to disinfectant performance within the facility. By the end of this presentation, you should have a clear framework for designing studies that are scientifically sound, operationally realistic, and aligned with regulatory expectation. Here's a quick overview of the topics we'll be covering today. We'll start with a few foundational definitions to make sure we're all aligned on terminology. Next, we'll walk through the indications for when disinfectant efficacy testing is required. This includes both the initial qualification phase as well as situations where requalification or additional testing becomes necessary. From there, we'll talk about how efficacy is determined, what criteria are used, how success is measured, and the scientific principles behind assessing microbial reduction. We'll then connect this to a clean room environmental monitoring program, which is one of your most important tools for designing a meaningful disinfectant study, so we'll discuss how that informs your testing strategy. After that, Shelby will give a brief explanation of the test method itself, what happens during a coupon study, what key variables are controlled, and how results are generated. We'll then move into study design, focusing on practical considerations that help ensure your study reflects real facility conditions and avoids common pitfalls. And finally, we'll wrap up with a discussion of what can happen if a study is not executed properly. These implications can include regulatory challenges, ineffective contamination control strategies, and the need for repeat studies. With that outline in mind, let's move to definitions. Let's review a few key terms you'll hear throughout the presentation. Environmental monitoring, or EM, is a systematic program we use to keep an eye on the microbial and particle conditions in controlled areas like clean rooms. The goal is to catch anything that could potentially contaminate your process. A sanitizing agent is any product that reduces the number of microbes on surfaces, including bacteria, viruses, and fungi. Think of it as a general cleaner that lowers microbial levels. A disinfectant agent goes a step further. It's designed to destroy or remove harmful microorganisms when applied to a surface, targeting vegetative forms specifically. Sporicidal agents are chemicals that can actually destroy bacterial and fungal spores when used at the right concentration for the right amount of time. So they're a bit more powerful than your standard disinfectant because they tackle the toughest forms of microbes. A coupon is a small piece of material that represents a surface in your facility. We use these in studies to test disinfectants in a controlled way without impacting the whole room. A neutralizer is a medium we use to stop a disinfectant from continuing to kill microbes at a certain point during the test. This ensures we can accurately count any surviving microorganisms. If your facility aseptically manufactures pharmaceutical products, the FDA expects you to validate that your cleaning and disinfecting procedures are effective. Even if your products are going to be terminally sterilized, it's still important to perform a risk assessment to make sure you're controlling microbes across the facility. In many cases, running a disinfectant efficacy study can be really beneficial to confirm your procedures are robust. For facilities under 21 CFR Part 820, which covers contamination control in medical device manufacturing, similar expectations apply. You need to demonstrate that your cleaning and disinfecting processes effectively manage microbial risk. In short, if your facility is making any critical, sterile, or sensitive products, disinfectant qualification is a regulatory expectation and a key part of keeping your products safe. From the disinfectant manufacturer, you want to see a clear certificate of analysis which confirms the product meets its specifications. This usually includes a chemical analysis of active ingredients, evidence of efficacy in liquid culture, and recommended contact times. On the end user side, your responsibilities include conducting an on-site audit of the disinfectant manufacturing facility to ensure quality and compliance, and of course, performing your own disinfectant efficacy testing in your facility is essential to confirm that the product actually works on your surfaces under real-world conditions. Basically, it's a partnership. Manufacturers provide baseline data, but the end user needs to verify that the disinfectant performs as expected in their specific environment. So what is the point of disinfectant efficacy? In short, it's to prove that your disinfectant procedures actually keep your manufacturing environment safe and free from harmful microbes. You might hear these tests called different things: coupon test, carrier test, or disinfectant qualification, but they all have the same goal, making sure your disinfectants work where they're supposed to. At Nelson Labs, we call it testing disinfectant agents for antimicrobial action on hard surfaces, or DET. The takeaway is this: it's not just about what works in the lab, it's about what works in your facility. At Nelson Labs, we perform disinfectant efficacy testing in alignment with USP 1072 and PDA Technical Report 70, both of which outline the fundamentals of effective cleaning and disinfection programs for aseptic manufacturing. But what really makes our approach stand out is how personalized it is. We don't just run a generic test, we use your actual surfaces and your materials. The image on this slide shows how we set up testing. You can see the individual surface as well as the neutralizer that has been qualified to stop disinfectant action after the desired contact time. We also have the capability to customize testing to match your internal procedures. So whether your facility has unique application methods, specific contact times, or specialized cleaning workflows, we can tailor the study to reflect exactly how your disinfectants are used in your facility. The result is data that's meaningful, applicable, and truly representative of your processes. Let's talk about when you actually need to perform disinfectant efficacy testing. A big one is when you have a new facility. Anytime you're building or updating a space to manufacture a new product, like adding clean rooms or other controlled areas, you want to verify that your disinfectants work on those specific surfaces. This type of testing is also helpful when you're developing or updating your facility's cleaning procedures, or if you've gone through a remodel and introduced new materials into your clean room. Another trigger is when the sponsor updates internal disinfecting procedures. This includes switching to a new disinfectant with different active ingredients, or when your environmental monitoring program starts detecting a new emerging contaminant. Those changes usually mean it's time to reassess your disinfectant's performance. And finally, disinfectant testing may also be required as a result of regulatory action. If the FDA finds gaps or weaknesses in a previous study, they may ask for additional testing to close those gaps. Anytime there's a significant change in your facility, your disinfectant, or your environmental flora, it's a good idea to evaluate whether your disinfectant is still working as intended. Here's an overview of disinfectant efficacy testing. First, we validate our organism recovery methods according to USP 1227. This ensures we can reliably recover microorganisms from the materials we're testing so the results are accurate. Next, we prepare the coupons or carriers, which represent the actual surfaces used in your facility. This is done by EO sterilization of each surface. A known concentration of challenge organisms is then inoculated onto each coupon. This gives us a consistent starting point for measuring microbial reduction. After that, we apply the disinfectant exactly as specified by the sponsor, meaning the same application method, same contact time your team uses in the facility. Once the contact time is lapsed, each coupon is placed into a neutralizing solution. This step is important because it stops the disinfectant from continuing to kill microbes beyond the allowed time. We then extract the coupon to recover any surviving microorganisms. A portion of that extract is plated or membrane filtered so we can enumerate the survivors. After incubation, we count the colonies, and from there we calculate the organism log reduction for each disinfectant on each surface. This gives a clear picture of how well your disinfectant performs under actual conditions. Now let's discuss how environmental monitoring, or EM, ties into disinfectant efficacy. Anytime humans are present in a controlled environment, some level of microbial contamination is unavoidable. Even with the best cleanroom design and the most careful operators, people naturally shed microorganisms. This is why a strong environmental monitoring program is so critical. It helps us understand what organisms are present, how often we're seeing them, and where they're showing up. There are a few key factors that go into building a solid EM program. First is a selection of the right growth media. This ensures the organisms you care about will be detected. Second is choosing appropriate culture conditions to support recovery. And third is establishing a well-designed sample plan with clearly defined sites throughout your clean room. If your contamination recovery ever exceeds the action limits set in your EM program, USP 1116 recommends taking corrective action. One of those corrective actions is revising your sanitation program, which can include reassessing your disinfectants, how they're applied, and how often they're used. So your EM program doesn't just track contamination, it also helps identify where disinfectant strategy needs to be updated or requalified. At Nelson Labs, we can assist with setting up your EM program, interpreting your collected data, and action plans to mitigate contamination. If you'd like more information on this, please feel free to contact me. My contact information will be on the last slide of this presentation. Now I'd like to turn the time over to Shelby, who will discuss designing a robust disinfectant efficacy study.
Thanks, Cassidy, for the introduction. I want to go over a few factors to consider when designing a disinfectant efficacy validation that may play a role in the log reduction you're attempting to achieve. Specifically, using environmental isolates, the type and concentration of the microorganism itself. The log reduction requirement may vary depending on the organism used within testing. Next, the antimicrobial agent you are using. It's important to understand that the concentration is very critical for certain disinfectants like phenolics, alcohols, peroxide-based solutions, where improper dilution leads to rapidly poor effectiveness. Internally, you're going to probably assign an open use by period or an open expiry period of the antimicrobial agent, and it's important to capture this information within your disinfectant validation. Ultimately, this will be present within the testing matrix table that I will cover towards the end of the presentation. The surface type is important, especially when considering the porosity or the texture of that surface. This may play a role in the disinfectant's ability to kill or inactivate microbes that may be shielded by the nature of the surface. The way you apply your disinfectants is considered in protocol development. It should mimic your internal disinfection procedures. This is intended to capture physical removal of an inoculum via a mop or a wipe, or maybe it's just a general assessment of the disinfectant action through a non-mechanical method like a spray. Lastly, contact time is an important variable when performing a disinfectant validation and should be accounting for the airflow in your environments. When applied, disinfectants should be saturated on the surface for a period of time. This is something that you might take from the disinfectant use instructions provided by the manufacturer. But in rooms with high airflow, disinfectant drying may occur before the proceduralized contact time. In that case, it would require additional application of the disinfectant. So make sure you're accounting for this. This next slide, I want to talk about procuring supplies for a disinfectant efficacy study. Really, one of the more time-consuming parts of this validation is going to be gathering your surfaces. The easiest time to procure these surfaces is while the facility is being built or adjusted, which is shown on the picture on the right. The facilities team within your company should have record of surface type and specificity. For the sake of testing, if you have no ability to attain coupons in-house, we have coupon procurement companies that may be used to obtain coupons. However, I want you to keep in mind it's important that the surface type from these procured coupon companies is identical to what you have internally. Overall, the size that's preferred for these coupons is a 1-inch by 3-inch coupon or a 2-inch by 2-inch coupon for our method. You'll need to provide disinfectants from your facility or the disinfectant vendor. These are obviously used to represent the agents that you use to disinfect your clean room and can be provided within concentrate form or ready-to-use formulation. You'll need to provide internal spray bottles or internal mops or wipes to apply the disinfectant. The mops or wipes can be provided in full, to which we will cut the size to mimic the size of the coupon for ease of use. Environmental isolates that are within your internal culture library can be provided via lyophilized pellet or a media plate or slant. As we move further into study design, I want to talk about organisms. specifically from your environmental monitoring program. Cassidy briefly mentioned the fundamentals of your EM program. So how do you translate this into a robust disinfectant validation? Really, you're just going to want to take one organism from the following groups at a minimum. For vegetative bacteria, use skin microbes like staph or micrococcus species. For yeast, an example would be Candida species. And then Aspergillus and Bacillus species would be a good example of spore-forming molds and bacteria, respectively. Important to understand that the scope of your disinfectants to be qualified, and you need to select your organisms appropriately. Keep this in mind. I'm going to touch on this later for the log reduction requirements on vegetative organisms and spore-forming organisms. So you may be asking, I have a new facility. What if my environmental monitoring data is not available for this new facility, but I still need to qualify my disinfectants? Testing may be performed with procured ATCC organisms or from another cell bank to determine disinfectant efficacy against a standard panel of gram-negative, gram-positive vegetative bacteria, vegetative yeast, spore-forming molds, and bacteria. As you see on the list, we have a good blend of E. coli and Pseudomonas for gram-negatives, gram-positives such as Staph aureus, Candida albicans for yeast, Penicillium aspergillus species for spore-forming molds, and Bacillus subtilis for a spore-forming bacteria. This next slide is going to dive into the surfaces. Understand that all surfaces are to be included within your clean room, such as your counters, your hood surfaces, your walls, your floors, critical equipment within this area, even the glove surfaces. If all surfaces are not included within the scope, justification needs to be provided to defend why you have limited the amount of surfaces included in the validation. To emphasize this point, I have added a 483 observation from the FDA that states directly, all surfaces that are used in critical processing and manufacturing areas were not evaluated in the disinfectant efficacy verification for hard surfaces. So as acquiring these coupon surfaces can be challenging, understand the importance of keeping these on hand internally. As we move forward with study design, it's obviously important to talk about the disinfectants themselves. A typical approach to decontaminating or cleaning the clean room involves a rotation of applying general disinfectants, sporicidal agents, and sanitizers to the surfaces. Examples may include 70% IPA as a sanitizer, pesticide as a general use disinfectant, and then sporicidal agents such as spore cleanse or decon spore. It's important to understand what surfaces are receiving which disinfectants, as this is going to play a role in developing the scope of your validation. Due to uncertainties in production and accessibility, even error internally, it may be beneficial to validate multiple agents from the categories I mentioned above. Historically, it's believed that you needed to rotate disinfectants to minimize microbial resistance to disinfectants over time. However, there is no real-world conclusive evidence indicating that this is true. To further drive this point home, I wanted to showcase some verbiage within PDA TR 70. This is showing that microbial resistance to disinfectants is based on a theoretical relationship to the resistance found within antibiotics. As you may know, antibiotic resistance is usually gained through modification of a single gene. Disinfectants are really going to have a variety of effects on cellular functions, meaning that development of resistance is not expected. But if you're interested, USP 1072 lays out nicely the classifications of disinfectants and their mechanisms of activity against microbial cells. So to reiterate, you may not need to rotate disinfectants because of microbial resistance. More so, rotation of your general use disinfectants, your sporicidal agents, will help combat the potential presence of microbial life within your clean room. So we've talked about the disinfectants themselves, but it's equally important to consider how you are applying them and to what surfaces they're being applied to. You may apply your disinfectant via spray in your clean room. As such, this is going to be replicated into your validation by spraying the coupon surface with inoculum present. This may be showcased by a droplet method, where we would directly pipette a disinfectant bubble onto the inoculum and allow contact time to elapse. For mechanical methods, you may apply a disinfectant through a saturated wipe or a mop, and this would be replicated into the validation by testing that mechanical removal of the inoculum and the chemical action of the disinfectant left over. Again, your mop or wipe would be cut to size of the coupon and is going to be performed through a single mechanical motion from top to bottom. Keep in mind that the application method of your disinfectants is really going to vary from surface to surface within your clean room. As such, you need to have your disinfectant validation matching the application method per surface. So this slide is going to go over an example disinfection efficacy testing matrix. This is going to determine the total testing arrangement of a new coupon study. This is expected to scale if you're validating your entire process, including all surfaces, all disinfectants, all potential organisms, or maybe you're just introducing a new surface to your clean room and need to qualify the disinfectants for the new material. Some things to notice on this slide. In the second column, notice which disinfectants are being tested against which microorganisms. Notice that the sporicidal agent, Spore Cleanse, on the bottom row includes testing against spore-forming molds and bacteria. The surfaces testing are varying based off which disinfectant is being used. This is matching how the cleanroom operator would apply the disinfectant per surface. The contact time and the application method of the disinfectant is varying based off your internal procedures or your process. Lastly, in the last column on the right, as you've likely assigned an open use by expiration of the disinfectant, it's important to understand that you're testing that disinfectant at a greater than or equal to expiry time. This is showing that you can still demonstrate the minimum log reduction requirement even when that disinfectant bottle is open for 14 or 30 days. Generally, this testing matrix is going to fully encompass the data you're needing to gather to support your internal disinfection methods. So with this information, it's good to understand that scope can expand very quickly, and so can the cost of this testing. So there's a few ways to reduce that cost, one of which reducing the contact time to only worst case. In this next slide, it's demonstrating the log reduction requirement that is present within the USP document 1072 and the PDA TR 70 recommended acceptance criteria. When you receive log reduction results, they're perfect for seeing if your processes are sufficient or not. So let's dive in. The first table lays out criteria within 1072. As shown, the values needed are a 2 log reduction of spore-forming organisms and a 3 log reduction of non-spore-forming organisms. Nelson likes to categorize vegetative bacteria and yeast within the non-spore-forming criteria. On the other hand, spore-forming bacteria like Bacillus species, spore-forming molds like Aspergillus would be categorized within the 2 log reduction criteria. The PDA recommends log reductions based on very minimal contact times, usually 1 to 5 minutes, and as such, that correlates to a greater than 1 log reduction. I do want to clarify really quickly that a 1 log reduction is a 90% reduction of a microbial population, a 2 log 99% reduction, and 3 log a 99.9% log reduction. To note here, rounding a log reduction to the acceptance criteria is not acceptable, meaning, for example, if you had a 2.65 log reduction and attempted to round it to 3, you're not representing a 99.9% reduction of the microbial population, and this is likely to face regulatory scrutiny. This slide's important to consider. It's going to cover how you should approach your acceptance criteria from a regulatory perspective. Your test design should be defined to align with acceptance criteria and regulatory expectations of reducing a microbial population. And in the second open bullet is an example of unrealistic criteria where it's being attempted to achieve a 3 log reduction of bacterial spores with 70% IPA sanitizer. So you want to look at your processes internally and determine the correct surface, organism, disinfectant combination while monitoring the contact times to ensure you're selecting proper acceptance criteria. I've included a few 483 observation examples from the FDA, the first of which is an example detailing the lack of validation, showing a disinfectant process that performs as expected. So make sure your validation is robust enough to cover your entire process. The next point showcases a validation that failed to meet log reduction criteria because recovery of the challenge organism was at a level that resulted in a lower log reduction on specific surfaces. As you may notice, that log reduction criteria is a bit higher than the industry guidance. So what I want you to gain from this is ensure you're selecting appropriate criteria that's going to showcase efficacy of your process. As we've talked about the evaluation of results and comparing them to acceptance criteria, I briefly want to revisit some common errors associated with this testing. This is going to get you into a design mindset that will help you proceed efficiently through this process. As I mentioned earlier, the most time-consuming portion of this validation will be obtaining surfaces. As a result, a common error will be not having all surfaces within your critical manufacturing environment represented within your DET validation. We just mentioned that in the acceptance criteria slide, it's important to understand that not meeting criteria or not defining the criteria well enough can be a major error. Next, be sure to include a broad range of either environmental isolates or ATCC procured organisms to represent the flora potentially present within your critical manufacturing environment. Even if you're providing data to a new surface within your clean room, you need to have a log reduction showing on both a panel of spore-forming bacteria, mold, then vegetative bacteria and yeast. Over time, it's recommended to revisit your current disinfectant validation and your EM data to determine if any newly identified species require additional testing or additional data. Lastly, the last bullet, disinfectant exposure times and the application of those disinfectants need to match your internal SOPs. So as such, make sure you're considering your open expiry dates as a part of your validation, like I discussed in the testing matrix slide. I want to talk quickly about our references and specifically mention that USP document 1072 was recently open to comments. This may result in adjustments to coupon size or maybe the method itself. So stay on your toes, be ready to adjust accordingly. If you have general inquiries regarding this testing, contact our sales team at the bolded email shown here. If you have any technical questions regarding this presentation or the testing, feel free to contact Cassidy and I at our information here. We're eager to act as an extension of your operations. We're hoping to help you develop a compliant disinfectant efficacy validation. Feel free to contact us regarding educational events and resources at Nelson Labs or any of the other business units. At this time, I'm going to turn it back over to Mike as we open the floor to question and answer. Thanks everyone for joining.
And thank you, Shelby and Cassidy, for this great presentation. We do have questions that are being submitted, and you're encouraged to continue to submit those questions, and we will try to answer as many as we can. If we don't get to your specific question, the team would be happy to address it individually after we conclude our webinar today. And again, joining Cassidy and Shelby as needed will be Tanya Morris and Aaron Zani and Michelle Lee. So let's go ahead and get into our first question. Our first question, Shelby, You talked about glove surfaces being involved in surface coupons. Was this for gloves that are integrated into cleanroom areas, or was it meaning the gloves used by the staff applying the disinfectant?
This is going to apply to isolator gloves specifically, maybe within a RABS unit or something similar. As well, the latex gloves being used to wipe down materials with disinfectant.
Okay, thank you. Here's another question. How many times can we reuse the coupons?
Hi, Mike. This is Aaron Zani. I'm just going to jump in on Shelby's computer here. So reusing coupons will be performed until they are no longer representative of the material. So during the testing process, if any damage occurs to the coupon surface, or if we see any swelling of any backing material, then that surface will no longer be used in subsequent testing. So often when we set up these validations, we're going to provide a number of coupon surfaces that allows testing to be performed without any reuse of the coupon. This is a preferred method as testing could cause additional wear and tear on that surface. While that does maintain a worst case approach, we don't want to get outside the bounds of what is representative of your facility. So when you are submitting samples and coupons for a disinfectant efficacy validation, we do encourage you to reach out to request how much do we need to perform this. And we'll provide that worst case number of coupons that allows us to not only complete the study, but to have extras in case we do have any retesting events.
Great, thank you, Aaron. Here's our next question, and I hope to pronounce this correctly. Albumin 0.3% can be used in disinfectant efficacy testing to simulate the loss of activity of the disinfectant due to organic matter on the surface. Is there any consideration to use albumin so it does not interfere with the recovery of microorganisms if evaluated?
This is a good question, Mike. And something I want to point out is that when we're performing disinfectant efficacy testing, we are assuming that this material has been cleaned. So, but when we're adding disinfectant, we want to make sure that that surface already had that organic material or residues from your process removed. When manufacturers of these disinfectants are performing their efficacy testing according to EPA requirements, often there is interfering substances employed, and that is to make sure those disinfectants will work in the field in a range of environments that may or may not be clean. However, in our aseptic processing facilities, we do want to have a cleaning step before we apply disinfectant. If there is specific processes that will not allow you to clean a surface before you apply your disinfectant for the appropriate contact time, we can work with you to create custom protocols that allow interfering substances to be included. And those processes of how we apply that organic material, how it's combined possibly with inoculum, those can all be tailored to meet your requirements and your needs of your facility. But we do want to stress that this chapter in the USP is about working with clean surfaces. So we do encourage that your processes first clean, remove these residues, and then we apply disinfectants to achieve the log reductions that we're looking for.
Great, thank you. Here is our next question. Is there a recommended time frame for revalidation even if surfaces, processes, and disinfectants have not changed?
Yeah, at this time, there's not really a recommendation with regards to revalidation if nothing has changed. However, I mean, over a long period of process, you know that your bioburden or your environmental monitoring and the bioburden that you're seeing in that environmental monitoring is going to change, maybe not significantly, but slightly. So I would probably take a look at your environmental monitoring, look at the trending, see if you're trending upwards in any particular organism, or whether you're seeing some microbial disturbances, even related to spores or fungi. I know during different seasons, sometimes that environmental monitoring could shift or change. I would evaluate those. And if you're seeing any of that, potentially look at revalidating or reassessing your disinfection validation. But currently, right now, there's not really any specification as far as redoing your validation other than any changes or anything to the qualification.
Great, thank you, Tanya. Here is our next question. The FDA 483 you cited is for hard surfaces. Are gloves, I think it's supposed to say, are gloves to be considered a hard surface?
Yeah, Mike. So this is one of those things where the guidance may not extend to what we see in regulatory questions and regulatory requests for additional testing. So while gloves are definitely not a hard surface, they are, they're going to be a variable topography surface. They are going to be challenging, but we often see regulators asking our sponsors for data on their gloves. So while the chapter is, specified for the hard surfaces like your steel, your floors, your walls, we do often see the gloves are part of that process, of that aseptic processing. And regulators want to know what is your either justification for not including those surfaces in your validation, or can you show me some data to show that your disinfectant is working? Previously, we talked about, what kind of gloves that should be included in these processes, right? Isolator gloves are very common. And if you're using sterile gloves, right, to apply your disinfectant, those may not be included in the validation if you show, well, these are sterile, they're aseptically, gowned, and they should not introduce any microbial content. But if your process leads to the possible introduction of microbes through the use of gloves, regulators may ask that you still include those in your validations.
Great, thank you. Here is our next question. The log reductions that I have obtained are not what I expected. What should I do now?
This is a good question. There are a few options you can consider to do following this. First, we can change the elements of the study. That's going to include longer contact times, alternative application methods, different concentrations of the disinfectant itself. But overall, investigational testing may be used to see if the result is producing an unexpected log reduction or is an outlier.
Okay, great. Here's another question. We may have covered this, but let's just reiterate it. What is the acceptance criteria? How much log reduction is acceptable?
Yeah, and that's coming from that acceptance criteria slide showing a USP 1072 2-log reduction of spore-forming organisms and a 3-log reduction of vegetative or non-spore-forming organisms.
Great, thank you. We have a few more questions. Why would I need to rotate disinfectants if a sporicidal agent is expected to kill everything?
This is also a good question. Typically, sporicidal agents themselves are going to be harsher chemicals. That means their application can be overall a bit more irritating when applying those disinfectants, so you can kind of tie in a safety aspect to this. But it also means they're going to be more damaging to the surfaces in the clean room itself. While applying a sporicide on like a regular schedule would be sufficient, it may impact the surface finish over time. So rotating in disinfectants, rotating in sanitizers that target vegetative life forms allow you to maintain control of your site and also help prevent degradation of the surfaces in the clean room.
Great, thank you. Here is another question here. You mentioned the importance of challenging application method as part of validation. What is the guidance or requirement for challenging this as part of DET or challenging in other validation activities such as in situation, etc.? If you need that repeated, I'm happy to repeat. It's kind of a long one.
Yeah, Mike, a repeat would be great here.
Yeah. So as you mentioned, the importance of challenging application method as part of validation. What is the guidance or requirement for challenging this as part of DET or challenging in other validation activities?
Hi, Mike. Yeah, this one is a complicated question, and I think that it requires that we take a look at the guidance a little more specifically, and also some of the new guidance that we're getting from, the EU Annex 1. So I think this would be a good question for us to respond to in an email and get a little more specific so that we can cite the specific regulatory comments.
Perfect. And we will reach out to you with that answer. Our next question is, should log reductions take grade level classification of environments into account or end sterilization step for medical devices?
So the log reduction is going to be based on the performance of the disinfectant on the surface. It's not really going to affect the grade level classification or, and it's not part of a sterilization step for a medical device. That would be a different type of test. So I hope I'm answering that. I'm answering your question. Or that I'm interpreting it correctly. But any sterilization step for a medical device, that's a completely different type of a test. That'll be a disinfection for medical device testing, and you're using a high-level disinfection in that case. So if I haven't answered your question, please let us know, and I will follow up in an email with that regard.
Great, thanks, Tanya. And we have one more question here. It says, what is the recommended number of replicates to perform this testing with?
Great question, Mike. Here at Nelson Labs, we recommend a minimum of triplicate to be used in testing for both positive controls and test articles. And then that's for every disinfectant organism surface combination. Since this test use custom surfaces, possibly custom application methods, we're really going to want to account for the variability that's inherent to the test. We can also perform this testing with more replicates if needed. Overall, if investigational testing is taken, additional replicates could be used to determine an outlier result if it's observed in the original testing run.
All right, great, thanks. Thank you for that. And we're coming up on time, and I want to make sure that that we've been able to get through everything and all of our questions here. So we do appreciate that you have participated in this webinar. And Nelson Labs does host educational seminars for medtech professionals to establish or refresh their fundamental testing knowledge and thereby achieve more efficient, accurate, and testing outcomes. And by attending this webinar, we'd like to offer you a discount for this 3-day seminar. If you visit nelsonlabs.com, under the News and Events section is our seminars page. You can use the code SEMINARS10 for this discount. I would like to thank our presenters, Cassidy and Shelby, for their insights, and I would like to thank Tanya and Aaron for their participation in the question and answer as well. We hope that you, the viewer, found this session informational and found it to be a valuable experience. And we hope that you have a great rest of your day.